यूनिट 6 रिप्रोडक्शन। इसमें हम लोग सेक्सुअल रिप्रोडक्शन इन फ्लावरिंग प्लांट्स पढ़ते हैं, ह्यूमन रिप्रोडक्शन पढ़ते हैं और रिप्रोडक्टिव हेल्थ पढ़ते हैं। इसका इंट्रोडक्शन पार्ट है। बायोलॉजी इन सेंस इज द स्टोरी ऑफ लाइफ ऑन अर्थ। विल इंडिविजुअल ऑर्गेनिजम्स डाई विदाउट fail? A species continue to live through millions of years unless threatened by natural or anthropogenic extinction. Reproduction becomes a vital process without which a species cannot survive for long. Each individual leaves its progeny by asexual or sexual means. Sexual mode of reproduction enables creation of new variants so that survival advantage is enhanced. This unit examines the general principles underlying reproductive processes in living organisms and then explains the details of this process in flowering plants and humans as easy to relate representative examples. A related perspective on human reproductive health and how reproductive ill health can be avoided is also presented to complete our understanding of biology of reproduction. अब हम लोग इसमें देखेंगे पंचानन महेश्वरी 1904 से 1966। Born in November 1904 in Jaipur, Rajasthan, Panchanan Maheshwari rose to become one of the most distinguished botanists not only of India but of the entire world. He moved to Allahabad for higher education, where he obtained his DSc during his college days. He was inspired by Dr. W. Durgeon, an American missionary teacher, to develop interest in botany and especially morphology. His teacher once expressed that if his student progresses ahead of him, it will give him a great satisfaction. These words encouraged Panchanan to inquire what he could do for his teacher in return. He worked on embryological aspects and popularized the use of embryological characters in taxonomy. He established the Department of Botany, University of Delhi as an important center of research in embryology and tissue culture. He also emphasized the need for initiation of work on artificial culture of immature embryos. These days, tissue culture has become a landmark in science. His work on test tube fertilization and intra-ovarian pollination won worldwide acclaim. He was honored the Fellowship of Royal Society of London (FRS), Indian National Science Academy and several other institutions of excellence. He encouraged general education and made a significant contribution to school education by his leadership in bringing about the very first textbooks of biology for higher education secondary schools published by NCERT 1964. अब हम लोग फर्स्ट चैप्टर में चलते हैं, सेक्सुअल रिप्रोडक्शन इन फ्लावरिंग प्लांट्स। इसमें 2.1 यूनिट है, 5 यूनिट है टॉपिक पढ़ना। 2.1 में है फ्लावर ए फैसिनेटिंग ऑर्गन ऑफ एंजियोस्पर्म, free fertilization structure and events, double fertilization, post fertilization structures and events, apomixis and polyembryony. Are we not lucky that plants reproduce sexually? The myriad of flowers that we enjoy gazing at, the scents and the perfumes that we shone over, the rich colors that attract us are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own selfishness. All flowering plants show sexual reproduction. A look at the diversity of structures of the inflorescence, flowers and the floral parts shows an amazing range of adaptation to ensure formation of the end products of sexual reproduction, the fruits and seeds. In this chapter, let us understand the morphology, structure and the processes of sexual reproduction in flowering plants and angiosperm. 2.1 Flower, a fascinating organ of angiosperms. Human beings have had intimate relationship with flowers since time immemorial. Flowers are objects of aesthetic, ornamental, social, religious, and cultural value. They have always been used as symbols for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. List at least 5 flowers of ornamental value that are commonly cultivated at homes and in gardens. Find out the names of 5 more flowers that are used in social and cultural celebration in your family. Have you heard of floriculture? What does it refer to? To a biologist, flowers are morphological and embryological marvels and the sites of sexual reproduction. In class 11, you have read the various parts of a flower. Figure 2.1 will help you recall the parts of a typical flower. Can you name the 2 parts in a flower in which the 2 most important units of sexual reproduction develop? 2.2 Pre-fertilization, structures and events. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated which lead to the differentiation and further development of the floral primordium. Inflorescences are formed which bear the floral buds and then the flowers. In the flower, the male and the female reproductive structures, the endoecium and the gynoecium, differentiate and develop. You would recollect that the endoecium consists of a whorl of stamens representing the male reproductive organ and the gynoecium represents the female reproductive organ. 2.2.1 Stamen, microsporangium and pollen grain. Figure 2.2a shows the two parts of a typical stamen, the long and slender stalk called the filament and the terminal generally bilobed structure called the anther. The proximal end of the filament is attached to the thalamus or the petal of the flower. The number and length of stamens are variable in flowers of different species. If you were to collect a stamen, each from 10 flowers, each from different species, and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissecting microscope and making neat diagrams would elucidate the range in shape and attachment of anthers in different flowers. A typical angiosperm anther is bilobed with each lobe having two theca, that is, they are dithecous. Figure 2.2b. Often a longitudinal groove runs lengthwise separating the theca. Let us understand the various types of tissues and their organization in the transverse section of an anther. Figure 2.3a. The bilobed nature of an anther is very distinct in the transverse section of the anther. The anther is a four-sided tetragonal structure consisting of four microsporangia located at the corners, two in each lobe. The microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of an anther and are packed with pollen grains. A structure of microsporangium. In a transverse section, a typical microsporangium appears near circular in outline. It is generally surrounded by four wall layers. Figure 2.3b. The epidermis, endothecium, middle layers and the tapetum. The outer three wall layers perform the function of protection and help in dehiscence of anther to release the pollen. The innermost wall layer is the tapetum. It nourishes the developing pollen grains. Cells of the tapetum possess dense cytoplasm and generally have more than one nucleus. Can you think of how tapetal cells could become binucleate? When the anther is young, a group of compactly arranged homogeneous cells called the asparaginous tissue occupies the center of each microsporangium. Microsporogenesis. As the anther develops, the cell of the asparaginous tissue undergo meiotic divisions to form microspore tetrads. What would be the ploidy of the cells of the tetrad? As each cell of the asparaginous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or microspore mother cell. The process of formation of microspores from a pollen mother cell PMC through meiosis is called microsporogenesis. The microspores as they are formed are arranged in a cluster of four cells. The microspore tetrad, figure 2.3a. As the anthers mature and dehydrate, the microspores dissociate from each other and develop into pollen grains, figure 2.3b. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of anther, figure 2.3c. Pollen grain. The pollen grains represent the male gametophytes. If you touch the open anther of hibiscus or any other flower, you would find deposition of yellowish powdery pollen grains on your fingers. Sprinkle these grains on a drop of water taken on a glass slide and observe under a microscope. You will really be amazed at the variety of architecture, sizes, shapes, colors, designs seen on the pollen grains from different species, figure 2.4. Pollen grains are generally spherical, measuring about 25 to 50 micrometer in diameter. It has a prominent two-layer wall. The hard outer layer called the exine, is made up of a sporopollenin, which is one of the most resistant organic material known. It can withstand high temperatures and strong acids and alkali. No enzyme that degrades a sporopollenin is so far known. Pollen grain exine has prominent aperture called germ pores, where a sporopollenin is absent. Pollen grains are well preserved as fossils because of the presence of a sporopollenin. The exine exhibits a fascinating array of patterns and designs. Why do you think the exine should be hard? What is the function of germ pore? The inner wall of the pollen grain is called the intine. It is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by a plasma membrane. When the pollen grain is mature, it contains two cells, the vegetative cell and the generative cell. Figure 2.5b. The vegetative cell is bigger, has abundant food reserve and a large irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and a nucleus. In over 60% of angiosperms, pollen grains are shed at this two-celled stage. In the remaining species, the generative cell divides mitotically to give rise to the two male gametes before pollen grains are shed three-celled stage. Pollen grains of many species cause severe allergies and bronchial afflictions in some people, often leading to chronic respiratory disorders, asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grass that came into India as a contaminant with imported wheat has become ubiquitous in occurrence and causes pollen allergy. Pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablets as food supplements. In Western countries, a large number of pollen products in the form of tablets and syrups are available in the market. Pollen consumption has been claimed to increase the performance of athletes and race horses. Figure 2.6. When once they are shed, pollen grains have to land on the stigma before they lose viability. If they have to bring about fertilization, how long do you think the pollen grains retain viability? The period for which pollen grains remain viable is highly variable and to some extent depends on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release, and in some members of Rosaceae, Leguminaceae and Solanaceae, they maintain viability for months. You may have heard of storing semen, sperms of many animals including humans, for artificial insemination. It is possible to store pollen grains of a large number of species for years in liquid nitrogen minus 196 degrees Celsius. Such a stored pollen can be used as pollen banks similar to seed banks in crop breeding programs.